Good day, grade 9 students, and welcome to our introduction video for our discussion for weeks 4 and 5. So our topic for this portion is the introduction to genetics, which you have an idea based on what you have learned during your grade 8 science with a focus on biology. So let us have a short review and recap, especially on how to use the Punnett square. As part of our enabling competency for weeks 4 and 5, let us have the simple introduction to genetics. Let us start with the different terms that you will need to remember for this particular part of our lesson. So the first term is heredity. So when we say heredity, it is the passing of traits from parents to the offspring. Another word that we need to remember is genetics. Basically, genetics is the study of heredity because it is the study of how traits are passed from parents to offspring. When we talk about heredity and genetics, it is essential that we mention the name of Gregor Mendel, which we dubbed or called as the father of genetics. It is because of his extensive study about pea pods that we have come to know about how traits are passed from parents to offspring. So, based on his studies, he has chosen two different pea plants. One that produces only green pods, and the other plant is the one that produces the yellow pods. He crossed this, so that means the pea pods become the parents, and he studied the offspring produced when he crossed these two very different type of plants. So with his experiments, he observed that some traits of the organism or the pea pods are dominant while the other traits are recessive. So when do we say that a trait is dominant or recessive? A dominant trait is a genetic factor that blocks another genetic factor. That means the trait is usually expressed, or in Tagalog, ito yung katangian na madalas lumalabas sa bawat henerasyon. While a recessive trait is a genetic factor that is blocked by the presence of the dominant factor. This trait is seldom expressed. The trait only occurs if both recessive alleles are present. Ito naman yung katangian na madalang lumabas sa bawat henerasyon. An individual that has a dominant allele and one recessive allele, the dominant trait will be expressed. So if you have the dominant allele, that means that dominant trait will be expressed. How are these traits identified? So let us trace the location of the gene in our chromosomes. During your grade 7 science, you have identified the different parts of the plant and animal cells. And one of the most important parts of those cells is the nucleus. It is within this nucleus that we can find the chromosome. The chromosome, in turn, is made up of our DNA, or the deoxyribonucleic acid. It is the genetic material that carries the different genes that are code for specific traits. So that means segments of that DNA are codes for certain traits. If we are going to have an example based on our illustration here, that specific segment of the DNA can be a code for, let us say, having curly hair. So that is how gene is located in our chromosomes. A gene with different information for a trait is called an allele. So it is represented by capital letter if it is dominant, and it is written in a small letter if it is recessive. Each chromosome pair has genes for the same trait, each one coming from each of our parents. So that means, For each gene, you have the trait coming from your father and the trait coming from your mother. So the combination of these traits will be your trait. The alleles of all the genes on an organism's chromosomes make up the organism's genotype. Basically, we can express alleles in two ways. The first one is by identifying the genotype. And when we say genotype, it is the trait written in letters. So we can have this as an example of the genotype for that specific trait. We can also have this one or even this one. So again, genotype is the expression of that alleles or the trait written as letters. Another way of expressing the trait is by the phenotype. This time, when we say phenotype, it is the description of the trait. So for example, you have red, you have white, you can have tall or short. So it is the description of the trait itself. So that is how we can express the trait or the alleles. When an organism's genotype has two identical alleles for a trait, it is called homozygous. Homo means the same or one. So it can be written as both capital letters. That means it can be read as homozygous dominant. Or it can be written as both small letters, identifying it as a homozygous recessive. An organism's genotype has two different alleles for a trait. It is called heterozygous. So if you have the combination of the dominant allele and the recessive allele, so you will have this as your genotype. It is called a heterozygous. Hetero meaning different. Based on Mendel's laws of inheritance, if you see this combination of letters, so the trait would still fall under the dominant trait because it still has or it contains, the genotype contains the dominant allele. So this one is read as heterozygous dominant. In studying genetics, it is important that you know how to use the Punnett square. Punnett square is a tool used by scientists to predict how the traits are passed from the parents to the offspring. Based on the name itself, it is drawn as a square divided into 4 sections. You can have the lines drawn up to those different corners, or you can actually extend those lines to help you write the alleles of the parents so as not to confuse them with the alleles formed through the inheritance patterns of the offsprings. So let us have an example of using this Punnett square. So our example trait is free earlobes. So it will be the dominant trait, free earlobes, which will have a genotype of both capital letter E. Our recessive trait is the attached earlobes, which will be represented by the genotype small letter E. So based on our example, let us draw our Punnett square and then write the numbers 1, 2, 3, and 4 to indicate the offspring. Let us have the following as the genotype of the parents. For the father, he is considered as the homozygous dominant. Again, it will be written as both capital letter E. And the mother is homozygous recessive. That means she has an attached earlobe, which is represented by the genotype small letter E's. So let us write those genotypes of the parents. We are going to write the genotype of the father at the top and the genotype of the mother on the side. So these genotypes are also called the alleles. So you have 2 alleles for each.
Filling out the Punnett square is actually very easy. So let us follow these simple steps. The first step is to copy the first allele coming from the father to this first section of your Punnett square. Next, let us copy the first allele coming from your mother on this first section. So you now have the genotype of the first offspring. To continue, again, let us copy the allele coming from the father and the allele coming from the mother. For the third and the fourth offspring, it will also be the same. So you have now filled out your Punnett square. The next step is to identify the genotype of the offspring. So to identify the genotypes of the offsprings, you are just going to copy those different letters as the genotypes. And for the phenotypes, you have the description. So you can see that the phenotype of the offspring are actually the same as the phenotype of the father, which is dominant, a free earlobe. Why is this so? Because as you can see, based on the letters, you have the capital letter E present, which represents the dominant allele. Based on Mendel's laws of inheritance, once the genotype contains the dominant allele, the dominant trait will be expressed by the offspring. So in our case, the dominant trait, which is the free earlobe, is the trait of the offspring. So all of them have the dominant allele, so they have the dominant trait. To finish this identification, let us have the genotypic ratio. So it is 100% heterozygous dominant. And for the phenotypic ratio, you have 100% free earlobes. So that is how you fill out the Punnett square and identify the genotype, phenotype, genotypic ratio, and the phenotypic ratio of the offspring from each parent. So hopefully you have learned how to fill out our Punnett square. So that is all for our review of the genetics. So hopefully you will learn from this portion so that for the succeeding part of our lesson, you will know how to fill out the Punnett square and also identify the dominant from the recessive traits and the difference between the genotype and the phenotype, as well as the difference between the homozygous and heterozygous traits.